I'm going to talk about a novel therapeutic that targets CCR4, a chemokine receptor, for the treatment of asthma and potentially other allergic and T2 high diseases. Let me see, where's my clicker? I've been working in this field for 30 years. I've had one job in my whole career, and this is it. But perhaps I will have another. Let's see, how do I move forward? This one? Okay, good. So here is the landscape, the market landscape. So asthma across the planet affects about 500 million people. Probably if you add in allergy and other T2 high diseases, we're talking about, potentially 1 billion people. In the US, the market's $60 billion to affects the healthcare system. And the most severe patients with the disease receive biologics is about 10% of the population. Now, standard therapy for asthma is inhaled drugs, and you can see that in the middle part of the slide. And then as we move up, companies have developed these incredible biologic drugs that are injectable, dosed monthly, but we don't have an oral agent to treat asthma or allergic diseases. So that's really our opportunity here. A small molecule oral agent that is actually an alternative to an inhaler, which I think everybody can get into. So there's a large unmet need. So here's the landscape you can see here. Our agent is a novel CCR4 antagonist up there in green, and you can see the other agents in the space. We have biologic drugs at the bottom and the inhaled therapies. And there is one other oral agent, montelukast, that's been developed over the years, but it's so weak that, in fact, many of my colleagues call it the best oral agent asthma drug placebo that ever got to the clinic. Sorry, I scrubbed that joke. All right, so this is the pathway. So CCR4 is expressed on T cells and other cell types, but it's a known validated target in inflammation. And when you inhibit it, you inhibit T2 inflammation. You can see the cytokines at the bottom, IL-4, 5, and 13 that are also the targets of biologics. Now here's some data from our lab. You can see single cell of the airway, and you can see the CCR4 is actually one of the things expressed between dendritic cells and T cells, that pathway that's activated in asthma on the left. You can see many different cell types and receptors being activated compared to controls. And on the right, on the top bar there, you can see the expression in asthma of the CCR4 pathway. Now, there was a drug developed by a company called Rapt Therapeutics that I've been working with in the last many years. That clinical trial, that drug made it to phase 2 clinical trial, but it was stopped due to toxicity in the atopic dermatitis trials. Now, we got 40 patients into that trial, and samples from that study are available and being shipped to Yale right now for us to do studies on if we get Blavatnik funding. But Rapt has developed a new asset, Rapt 847, that's been de-risked, has the same target and more potency. Here's data from the clinical trial, the phase 2. You can see here the treatment with the drug increasing lung function compared to placebo after inhaled steroids are withdrawn. Here's our target product profile. You can see here we believe our asset is going to be in the ideal position here with oral administration, high efficacy across the spectrum of disease, and is an alternative to inhaled steroids with minimal toxicity. So here's our team. GSK bought Rapt Therapeutics and is out-licensing this asset to the team, which includes at this point me and former Rapt employees, Dirk Brockstead and then Brian Wong, who was the CEO, who helped to lead Rapt to being sold to GSK. So we'll use the funds to do statistical analyses and assays on the samples from the phase 2 trial. So let me stop there and take questions. Thank you.
Great, thank you.
As you consider the evolving standard of care with TSLP and IL-13 antibodies, and both alone and in combination, and with companies developing YT mutations for bispecific antibodies, where do you feel that this fits in terms of this evolving standard of care by the time you actually...
Yeah, well, I think that it will be an add-on either way. So remember, these drugs, even the long-acting YTE modified biologics, are for the most severe patients. Clinical trials are being done looking at exacerbation reduction, not at milder disease endpoints or as they're add-ons to inhaled steroids. So this agent would be across the spectrum of disease. I would target the clinical trials at the mild to moderate patients first and not actually compete with the biologics to start with. But then, of course, they could be add-on to those as well.
I had a just quick question to clarify. So the asset that was in clinical trials that was discontinued due to safety reasons or adverse side effects, the second generation molecule, is that just next generation, or is it a novel compound, or is it just an iteration?
Yeah, it's a novel compound, screened at wrapped. The first drug, there was hepatic toxicity in one patient, and the trial was stopped. So when they developed RPT-847, it was de-risked, especially for liver toxicity. So they did a number of hepatotoxicity assays, and they did this DILI-SIM, drug-induced liver injury assay and simulation, to really kind of reduce the risk that there was any evidence of toxicity.
He deserves extra time. Okay, so I just want to follow on kind of the same line of questioning. So just so I understand the order here. So first there was 193, and there was toxicity issues on that. Then there was 847, of which you're getting the data for, and you want to use the grant to analyze the data of 847. Is 847 what you're looking to move forward, or is CCR4 a separate compound that you're going to be creating the new co around? That's where I'm a little...
Yes. So 193 was taken to phase 2. Okay, so that's where the data comes from, and that's where the samples come from. So that's serving as a model because we have phase 2 human data and human samples that we can analyze to bolster our case that 847 is going to be successful. 847 is IND ready at this point. Exactly. Yes. Well, it's negotiating with GSK